also write as PA and Staphylococcus aureus for all of these three. So you label at the back, you name your batch number and the date. You will be labeled them. EC is E. coli, PA Staphylococcus, Pseudomonas aeruginosa, and SA Staphylococcus aureus. Then use loop to do a very simple streak, just like that. But remember, how are you going to do this? Okay, you have a loop. Okay, the loop has to be flamed. You get all these labeled. You get the E. coli first, so you can do E. coli streak, E. coli streak, and E. coli streak. Then you have to flame it to get Pseudomonas aeruginosa. Then do Pseudomonas, Pseudomonas, and Pseudomonas. Then you flame it, you get Staphylococcus aureus, then do Staphylococcus, Staphylococcus, and Staphylococcus. So very simple. The idea is we're going to put the bacteria there. Then all these three plates, we do 35 degrees Celsius incubation, 24 hours. We will be explain the growth situation on these three media, which means we enrich differentiated media, C alpha, beta, gamma hemolytic, Macanchi agar selective differentiated media, mannitol salt agar selective and differentiated media. Okay, so this is the first work to do. Very simple. Okay, now second job we will to do. We will to see the external impact, or we say environmental factors for the bacterial growth. Usually the environmental factors for the bacterial growth, so we say external factor for bacterial growth. This including temperature. So that's why you have a thermophiles, mesophiles, hyperthermophiles, thermophiles, all these. The second one is for the pH, is that right? Acidic, neutral, and alkaline. Number 3 is oxygen. We have facultative, aerobic, anaerobic. And number 4 is osmotic. Osmotic. We already mentioned about that. Here, we're not going to do all these three. Osmotic, we will anyway. We will do on mannitol salt agar, so we assuming we already did. In your fact sheets, I wrote a lot, which is let you study, and we have exercises 3 and 4. Just give you an example. We will do exercises 1 and 2. Okay, it's very simple. So here we're going to focus on the temperature growing, temperature impacts for the bacterial growth. So how to do it? There is 2 work to do. Number 1, everybody will get 4 slants. Each of them you're going to have to label. Same thing you label right on the side. But very important, you're going to label the temperature. This will be labeled for C, 25C, 35C, and 55C. Then you will be inoculate, do a streak, a zigzag streak of your assigned culture. So let's do assignment. You guys will do E. coli. You guys do Staphylococcus aureus. You guys will do Pseudomonas. You guys will do Bacillus. Only one assigned culture, we will share the results, okay? So you will do either Staphylococcus aureus, E. coli, Bacillus, or Pseudomonas aeruginosa. Only one, okay, assigned culture. Then we give to the TA, we will incubate in a different temperature. The second one, we wanted to talk about this very special bacteria called Serratia. Okay, that bacteria will be right here called Serratia. This bacteria is very interesting. Usually the bacteria grow very well at 35, better than 25 because of temperature, because of the enzyme, and the good activity at 35. This bacteria will show you red to pink color at 25 degrees Celsius. And at 35 degrees Celsius, it is actually colorless. So this bacteria is kind of interesting. And this could be using a tracing marker in some of the purposes, for some of the purposes. So I want you to do a practice, which means everybody gonna get 2 slants. Both of them, we will be inoculate Serratia. Serratia, you can write SN, that's Serratia. One is at 25 degrees Celsius, another one will be 35 degrees Celsius. After growing, we will see the difference. Okay, so that's going to be the work you will do. So what's going to be the work today? Everybody going to come in here. It's very simple compared to what you did before. You will get 1 Macanchi agar, 1 mannitol salt agar, and 1 blood agar. Now, this looks similar. Is that right? This looks very similar, these two. So what you should do, hold on like this. You will see the edge very clearly. This is orange, red, this is purple. If you see like this, looks very similar when you first time see it. So you may get confused. You're not going to confuse the blood agar because it's very red. Okay? So that's something I want to mention. Now the second thing. So this is the first thing you do. Turn that. So the second thing, you will get the neutron agar slant right here. You will get 4 for the impact of the temperature growth, which will be inoculated to assign the culture. Strict plating of the slant, do a zigzag marker. And then you get another 2, which is for Serratia. One at 25, another one at 35. So how many slants you will get is 6. You get 6 altogether because it's all the same. Okay? So 3 plates, 6 slants. And go ahead to get it. All the agar plates, we have Staphylococcus aureus, Acidomonas aeruginosa, Bacillus, E. coli, and Serratia. It's all right here and also in front of your bench. Okay, so go ahead to do. Now don't forget, when you get everything done, count your results from last Tuesday. Then record it. Okay? That's good.